Welcome to Week 9, Lecture 2 of EBME401. This is the last lecture of the signals portion, and I've really enjoyed teaching you. I hope you feel like you've learned something and you've had a good time during this portion of the course as well. In this last lecture, we're going to take a look at image segmentation. There's also going to be a reading portion on image registration, which we'll briefly touch upon, which I would encourage you to look at. In addition to the spectral analysis and the filtering that we looked at in previous lectures and text portions, information can be extracted directly from images. So here we don't need to move to the frequency domain. We're just going to be pulling information out from the images themselves. So image segmentation is one operation we might want to do. So here we're separating an image into two or more distinct regions so that we can calculate information about those regions independently. We also might want to perform image registration. So here we're trying to align features in an image or to align two separate images in order to better compare the information in those two images. So both of these operations can be performed manually, but we would prefer them to be automatic because they might be kind of a pain to do manually every single time we're looking at these images. All right, so for instance, why might we want to perform segmentation, either manually or automatically? So one common reason that we want to segment images is so that we can define specific volumes. So an example that we'll be looking at a lot is calculating a blood pool volume in cardiac imaging. We want to know how much blood is in the left ventricle. So here we're trying to calculate an area in a two-dimensional image. And we may want to do this over time so that we can find out how much blood is being ejected from the heart in every heartbeat. And so here's an example where if we have multiple time-resolved images, we might want to have some sort of automatic way of performing the segmentation so that the physician doesn't have to go in and draw curves around the left ventricle at every different time frame of the images that we've collected to define this volume. So it'd just be way too much work for a physician. We might also want to define a volume so that we can calculate the size of a tumor or a lesion. So for instance, to see if a tumor would be dangerous at its current size or whether it's growing if we have multiple views of this tumor over time. We might also want to perform segmentation to mask out some unwanted portions of the image. If we have a very bright section of the image which is making it difficult to see other features, we might mask that out so that we can focus in on areas of the image that we're interested in looking at. We could also perform segmentation for surgical planning. So if we know we need to get, for instance, to one area of the brain, but we don't want to affect any other portions of the brain, then we can segment out exactly those portions and say we're not allowed to touch when we're doing surgery. A final example, for instance, if we're looking at pathology slides, we might want to count some number of features like the number of cells or the number of nuclei in order to get a handle on exactly what's going on in that particular slide. This is a particular tedious task. You may not want to sit there again and look through the slide and count the number of cells, and so it would be great if we could have some sort of automatic process which would allow us to do that. So let's take a look at a specific image segmentation example. So here what I want to do is separate the land, or the reefs, from the water. And this is pretty easy to do by hand. We see I have this patch of land here. Draw this in. It looks something like this. And I have a little reef over here. Here's all my land. And then I have all of this patch out here where it's also reef. So again, this might be kind of tedious, but I can certainly do this by hand if I wanted to. But imagine it's taking me too long, and so I want to ask a computer to do this. I can design an algorithm which would say, everything that's a specific intensity or color is land, and everything else is water. But I may run into trouble if I try to make some general rules. So for instance, there are some reefs that are underwater. They aren't actually land in my definition, but an algorithm that I would write might define them as land because their intensities are too similar to the actual land portions. And so I may get some errors in my segmentation if I use such a simple way of classifying land and water. I also may run into problems because there's two different kinds of water in this example. So we have some deeper water, which is darker blue, and some more shallow water, which is lighter blue. And depending on how I train my algorithm to separate land from water, it may have trouble with these two different kinds of water with different intensities. I also have these funny shadows that are near the land. And again, I know what I'm looking at, and so I know that this is probably some portion of the reef that's underwater, but to an image segmentation algorithm, it may be difficult to distinguish these shadows from actual land. So the point that I'm trying to make here is that segmentation might be an easy task for the eye, easy for me to do manually, but it might be something that a computer has a pretty difficult time doing unless we make a very specific algorithm. So we might have an algorithm that might work perfectly for this image, but then we throw in another image of a set of reefs, and our algorithm might completely fail to separate the land from the water in a new image because we had to make it so specific in order to get rid of all these special cases in this particular image. All right, so let's move away from our reef example and look at a medical image. So here again is an image of the heart. We've seen something similar. And imagine we want to figure out how much blood is being ejected from the heart with every heartbeat. So in that case, what we'll want to do is draw a contour around the left ventricle, so exactly the blood pool, and figure out what the area of this portion is. I'm going to erase this. So one thing that we have to keep in mind is that we might have some strange signals due to papillary muscles. We might want to exclude that, something like this. And so our contour might not be quite as nice, but we can still draw this pretty easily by hand. You can see where it might be difficult to train a computer to draw exactly this contour. So here is another example from a pathology slide. So here, we say we want to count all of these cells. Again, this may be very, very challenging to do by hand, and also very challenging to do with a computer, right? Like some of these have a very high intensity, but some of these are a little bit faded. Is that a cell or not a cell? How do you know if it should be counted or not? We'll have to design our algorithm very carefully. All right, and this is an example of an MRI image of the head, which we're going to use in a future example. So here we might want to define a contour all around the head in order to mask out noise. So get rid of all of these portions while still retaining these low intensity portions which actually do belong to the head. Similarly here under the skull. All right, so these are all different examples of times that we would like to have some sort of automatic segmentation, although it may be challenging to design an algorithm that does this. So when we perform our segmentation, the first task is usually to find edges or boundaries in an image, right? These edges are what's going to tell us what belongs to one segment and what belongs to another segment. So we can use one or a number of combinations of characteristics of an image. So in our reef case, we used color to tell us the difference between land and water. And this color is really just another way of looking at image intensity or magnitude. So we can also use, again, just the intensity or magnitude. We can use nonlinear filters to help us see texture in images. And we're going to talk about this textural analysis in a couple of slides. So once we know what kind of characteristics we want to use to find these edges, we can perform our edge detection or segmentation in a variety of ways. So what we're going to focus on is thresholding using a histogram analysis of pixel values. So this is what we'll be looking at for most of the rest of the talk. We can also use specific filters like high pass filters or finite difference filters or special canny or nonlinear filters in order to find specifically the edges in an image. And this you've seen in some of the text portions. There's also a lot of other different algorithms, including region growing, clustering, and model-based segmentation, which we aren't going to talk about. But these are active areas of research because people are still working on different methods of segmentation, which can be used to automatically process our images so that we don't have to go through them one by one. So let's take a look at, again, a very simple segmentation example. So what we want to do is separate pixels in the brain from background pixels. All right, so in other words, we're trying to draw this contour all around our brain. Something like this. And say everything within this contour, all of these pixels, this all belongs to the brain. And everything outside of this contour This is all background. So when we saw this image, when we first saw this image, we saw that if we just use intensities to define background versus actual pixels in the brain, we'd run into some problems. Right? Here we have low intensity values. This is clearly background. But there are portions of the image that belong in the head that also have low signal intensities. And so just performing some sort of thresholding operation isn't going to get us there. Let's take a look at how this would work. All right, so again, if we specifically look at a threshold operation, what we're going to do is say that all pixels above a certain intensity value belong to a segment, and all the pixels below the intensity value belong to a different segment. And we can make a binary mask of these two segments in order to help us extract one segment from the other segment. Okay, so in the specific example of the brain, we see that the pixel values range from 0 to 800. And so here I've just arbitrarily chosen a pixel value, 250. And what I've made here is a binary mask which shows only the pixels which have intensity values above 250. So essentially these are the pixels that appear bright in my image over here. So we see that this is a binary mask. It is performing some sort of segmentation, but we're not actually segmenting out any parts of the image that are particularly interesting to us. So how can we figure out what an interesting threshold value would be? Something that would start to separate these background pixels from the actual pixels showing tissues. And one good way to start selecting a threshold value is to perform a histogram analysis. So we take all of our pixels and we calculate how many pixels fall into different intensity bins. So here, we can see that the pixel values range from 0 to 800, and on the y-axis, we can see how many pixels fall into each of these little bins. What I then can do is look to see if there's any natural breaking point between different groups of pixel bins. And so for instance, we see that we have some shape that looks kind of like this. We draw an envelope over our pixel bins, and it looks like one natural place to draw a distinction between pixel bins is about at a threshold of 100. All right, so here's my threshold. And what I'll do is say that all the pixels that have a higher intensity will belong to one segment, and all the pixels that have a lower intensity, they'll belong to a different segment. Alright, so if I use this threshold value, we see that I'm actually getting a lot more of my image pixels than I did when I just arbitrarily chose this threshold to be 250. So looking at this histogram has given me a lot of information about which pixels might actually be interesting and which pixels I can just really throw away. However, we see that we're still having the same problem, that there's lots of areas of my image here and down here, which have low intensity values. Right? Those are pixels which are in the brain. Right? My brain is continuous, it doesn't have big holes in it. And so I want to somehow capture these pixels because I haven't really properly segmented my image until those pixels appear in my binary mask, showing the segment which corresponds to the head. So my goal is to get rid of all of these gaps that I have in the mask that I've made of my head. And one way I can do this is to use what are known as morphological operations. So these are nonlinear operations which enable the closing of gaps or the removal of spurious pixels that are appearing in my mask. So let's take a look at how we can do each of these. So one thing I can do is take all of the low intensity values of my mask, that's all the zeros or all of the blue areas in this image to the center, and I can grow all of those blue areas by 1 pixel. Right? So what we see is that here, for instance, this blue patch is pretty small, and if I erode by 1 pixel, I'm going to grow all of these blue areas. We'll see that this area is going to increase. And at the same time, all of these blue pixels are going to start encroaching on this red pixel here. So I may have a large box here, which will be slightly smaller. Right? And if I do the same thing to this pixel up here, we'll see that all of this blue is going to start encroaching on the red. and this little area is going to be smaller. It's going to be eroded by 1 pixel. And you can imagine that if I would erode enough, I could completely remove this pixel from my mask eventually. What I want to do though with my brain mask is exactly the opposite. I don't want to get rid of spurious pixels, but I want to fill in some gaps. And so in this case, I could use a dilation operation. So I'm dilating all of the red regions now by 1 pixel. So instead of getting rid of areas of my mask, I'm going to add to the areas of my mask. We'll draw this in yellow here. So again, I'm taking this red area, and everywhere where I have red, I'm increasing the red area by a pixel. So this is growing slightly larger. And again, my red area is here going to move into this blue area, making this gap here a little bit smaller. That's exactly what I want to have happen in my brain mask. In the spurious pixel, I have something happening that I don't want to have happen. Something that's small is actually going to get slightly larger here. But I could take care of that by first eroding and then by dilating. Alright, so let's take a look at how dilation and erosion will affect the masks that I have of my brain. Alright, so again, I picked a threshold based on a histogram, and after my thresholding, I have this mask here. And we see that the mask is okay, but I need to fill in these gaps. So if I dilate this mask by 1 pixel, so if I dilate this mask by several pixels, the result will be this new mask over here, where you can see that a lot of these gaps have been filled in. And if I dilate by an even larger amount, I can get rid of these residual gaps that I have over here. So eventually I will have filled in all of these areas in my mask. On the right-hand side, I have an example of erosion. So this spurious pixel didn't appear in my mask, but I put it in for this example. And if I erode this mask by a couple of pixels, then we'll see that, again, all of the zeros in my mask are going to grow and start eating into all of the values of 1 in my mask. And so I can essentially make that spurious pixel disappear by allowing the zeros to eat it away, essentially. So we see here, I'll also have the same effect in all of these areas here, which I don't necessarily want. But at least I've gotten rid of this spurious pixel here, and I could then dilate this image in order to eventually get back to something that looked like this. Right? So this would help me get rid of all those extra pixels, and then again go back and fill in those gaps. As we saw in the previous slide, it might be a benefit to perform one morphological operation followed by another morphological operation. And these dual operations actually also have names. So for instance, an opening operation is an erosion followed by a dilation, and a closing operation is a dilation followed by an erosion. So let's take a look at an example. So here I'm starting just with my thresholded image, and we remember that the goal is to get rid of all of these gaps where I actually have some brain tissue, but they're just appearing as dark in my mask because they have a low signal intensity. So what I can do is first dilate my mask, which is going to allow me to fill some of these gaps. We know whenever we do a dilation, we're growing our white region, so it'll be a little bit wider than it was originally. So I actually have now an outline here, which is a little bit wider than my original head. And so if I use this as my mask, I'm going to be retaining some of these background pixels, which are very close to my actual head, but I might not want these. So if I then take this dilated image and erode it, then this whole operation in total, so first dilation, then erosion, is equal to a closing operation. And so my closing operation will then take these image pixels and shrink them back down so that I should end up with a boundary of my head, for instance here, which is the same as this boundary of the head over here. But now I've gotten rid of all of the gaps in the middle of my mask that I wanted to get rid of. And so if I subtract my dilated mask from the one that's completely closed, we'll see that there actually is a difference. So this red region here is the part that's actually the brain, and this green portion here is what I would have had if I had just done my dilation, where I have, again, this edge of pixels which would be background signal that I don't actually want in my mask, but we've included it because of the dilation operation. So by going back and performing an erosion after the dilation, or a complete closing operation, I'm filling in the gaps, but still maintaining the actual edge of my brain, which is what I really want in my mask. Alright, so we see that after we've performed this closing operation, we're left with a mask that no longer has holes in it due to low signal intensities in the middle of the brain, but reflects the true border of my actual head. And so I can now use this mask and multiply it pixel by pixel by my original image in order to get a new segmented image that has gotten rid of all of the background information. And so here's my original image. Here's my segmented image. It's actually a little bit difficult to see the difference between these images, so I'm just going to scale them up. Alright, so in the scaled version we can clearly see all of this background noise here, which we've completely eliminated by multiplying this original image by the mask that we've developed. So if we recall exactly what we did, we first made a histogram of all of the pixel values in order to find a threshold value which would allow us to separate the background noise from the actual signal in the brain. We then created a binary mask, just made up of zeros and ones using the threshold value. And then we cleaned this mask up by using a closing operation, which allowed us to fill in any gaps while retaining the edges of our images. So this is a very simple segmentation procedure. We can see how it's worked to actually get us the information that we wanted in our brain. Alright, so let's take a brief switch here now and look at edge detection. So we've already seen how filters can be used to help us detect images. So for instance, I can use a Sobel filter, which will allow me to go from an original image here on the left hand side to an image which just contains horizontal edges. So here I've applied my filter in the image domain, and again I could apply that in K-space just to get rid of all of the low frequency information and retain high frequency information which corresponds to just horizontal edges. I can also apply two separate Sobel filters, one in the horizontal direction and one in the vertical direction, which will allow me to keep both horizontal and vertical edges. So I can use these edges again to help me perform some segmentation. So this was an example of how we could apply linear filters for segmentation. But let's turn to an example where we're looking at a nonlinear filter that we'll use for segmentation. So sometimes just looking at intensities won't work for edge detection or segmentation. So this example here that you're seeing on the right hand side is a good example where we can't use a histogram analysis, and we'll see that on the next slide. So we see that there are two segments in this image, right? There's this parabola here, so this segment up on top, and then a separate segment on the bottom, which I'm shading in red. So our eye can clearly distinguish between these two segments. But when I've made these segments, I've used the same mean value, but a different standard deviation value for each of the two noises in these two segments. And so if I actually look at the means of these areas, they're going to be exactly the same. But there's clearly something different about them. And what's actually different about these segments is their textures. So I have grainier noise in one of the segments and less grainy noise in the other segment. I know this is a very qualitative way of describing it, but this is something that my eye can see, but it's difficult to describe if I'm just looking at the means of pixel values. Right, so I said before that a histogram analysis isn't going to allow us to determine an appropriate threshold for this image here on the left. And we'll see exactly what I mean. So if I take my image and I make a histogram of all the pixel values, exactly as I did for my brain image before, we see this histogram just has one very broad envelope in it. All right, so my values are running here from 0 to 1, and I have 100 pixels with a value of 0 approximately, maybe 50 with a value of 1 over here, but there's no clear separation between these two different segments like there was in our brain image. So here there's no really clear threshold, and any threshold that I select will lead to an inaccurate segmentation of different areas. I'll just end up with different patches from each area, but not a clear distinction between the two. So what I've done is I've thought about why these two segments look different. There are differences between these different regions, and I know that there are differences in the standard deviations of the noise here. And so one way to separate these two segments would be to actually calculate the standard deviation over different patches of data in the two regions and use those standard deviations in order to make a histogram. So looking at the standard deviation of a patch of pixels is basically a 2D nonlinear filter, right? I'm taking a set of pixels, calculating their standard deviation, and that is my output, right? So this is essentially the same as the nonlinear filters that we've talked about previously. It's just in 2 dimensions. So all I have to do to make it a 2-dimensional nonlinear filter is just specify the block size that I want to use, or over which I want to filter in 2 dimensions. Okay, so what I've done is I've taken this image on the left-hand side, and I've calculated the standard deviation over a 3x3 block. So essentially I've taken a 3x3 group of pixels here, just schematically. I've calculated the standard deviation, and I've used that as my output for 1 pixel here. I then slide my block over by a set of pixels, calculate the new standard deviation, and that's the output over here. And I just slide this block all through my input image in order to get my output image. And we can see that these two regions now, if I calculate a histogram, have completely different means, right? So it's now clear that there's one area here where the standard deviation has a much lower mean. That's this region over here. And then a whole other area which has a much higher mean. And that's this region up here. And so I can now define an appropriate threshold which will allow me to separate these segments. So for instance, I could draw my threshold almost at 0.5, and this should theoretically enable me to tell the difference between these two areas. Alright, so we see that I can now draw this threshold automatically, and I have a mask here for the bottom area and a mask here for the top area. And so again, these two binary masks, if I multiply this mask by my original image, then I'll end up keeping all of these pixels in the lower area and segmenting out all of the pixels in the upper area. Alright, so we've seen a couple of different examples of how we can segment images. We've looked at histogram analysis, determining thresholds and morphological operations, and then applying linear or nonlinear filters. But let's go back to our very original example. So what we wanted to do was to calculate the blood pool area, so to figure out how much blood is contained in the left ventricle. We might also want to perform an additional segmentation where we're looking at the myocardial mass. So figuring out actually how many pixels and what this area is between the red curve and the green curve. This will tell us the mass of the myocardium. However, in this particular image, we see that performing the segmentation will be very challenging. If we try to use image intensities to separate signals, we may have contributions from the blood pool here, but also contributions from this blood pool here, perhaps contributions from tissues in the abdomen. This is up here, some fat, and we may not want those contributions. So just performing a threshold probably isn't going to get the job done. Even if we can perform thresholding, we have problems where our myocardium has a similar signal intensity to our papillary muscles. And we may not want to include our papillary muscles either in the blood pool or in the calculation of the myocardial mass. So these other features are going to distract from the actual segmentation that we want to do. And so, for instance, in this particular case, if you were a cardiologist or a radiologist and you wanted to find these measurements, you would do a lot of this manually still, just because these images are so difficult to segment automatically. There may be some human interface with a computer segmentation, but you still have to look at the segmentation before you could determine that it was actually correct. And so what I want to show here is that a lot of segmentation is still really performed by hand. And so this is an area of active research where people are looking at better ways for us to segment images, sometimes using intensities, but sometimes also using textures to better understand what the characteristics of these different image properties are. So with that, I'd like to wrap up the final lecture of the signals portion of 401. And again, it's been a pleasure working with you. I hope you've learned a lot, and have a great time in part 3.